Okay, so have you ever, stopped and thought about aging? Like, if it's just this inevitable wearing down of everything, or if there's something more going on, something deeper in our cells?
You're hitting on a really fundamental question there, and that's exactly what's driving so much research into aging these days. Like, what is it really? And is it something we can change?
And that leads us right into what we're diving deep into today.
Yep, the information theory of aging. Or ITOA.
ITOA, right.
Yeah.
It almost makes you think of, corrupted code or something.
Yeah.
But for everyone listening, we're going to break this down. I know you all love to really understand these complex ideas, but without all the crazy jargon. So that's what we're doing today. We're going to look at what this theory is all about and why everyone's making such a fuss about it. Sound good?
Sounds like a plan. We'll start by laying out the core concept of the ITOA. Then we'll dig into the evidence, especially the research coming from David Sinclair's lab. And of course, we have to look at the other side of the coin too. We'll be exploring some pretty compelling counter-arguments from folks like Aubrey de Grey and Charles Brenner.
Totally. I'm excited to get into all of that. But first, just to be clear, our main aim today is to really get a handle on this ITOA thing. What is it actually saying? What does the science, the actual research say, both for and against it?
Right. And the other important point here is to keep in mind this is a hot topic in science right now. It's a dynamic debate, not a done deal. So we're going for that balanced view, just sticking to the information as it's presented in the research, okay?
Okay, great. So let's unpack this, shall we? What exactly is the information theory of aging at its heart? What's the gist?
The core idea is this. ITOA proposes that what really drives aging is basically this gradual loss of information, youthful epigenetic information that's happening inside our cells. It's kind of, think of your DNA as the instruction manual, right? The fixed blueprint.
Mm-hmm.
But then you've got the epigenome, which is like all the highlights and notes and dog-eared pages on that manual.
Interesting. So the DNA is the blueprint, but the epigenome is how that blueprint gets used.
Exactly. It's telling the cell, hey, read this part or pay close attention here. But here's the thing. These epigenetic notes, they can fade, get messed up over time. And that's what the ITOA is saying is the real driver behind aging.
So it's like the software getting corrupted even though the hardware's still okay?
Yeah. That's a great way to think about it. The software here is the epigenome. It's all these chemical tags on the DNA and the proteins that package it up. They're what control which genes get switched on or off. And because it's so dynamic, the epigenome is much more sensitive to, noise. You could say that all the environmental factors and just general wear and tear on the cells over a lifetime, all that contributes to this buildup of noise. And that, according to ITOA, is the main culprit for aging.
Fascinating. And I was reading that this idea isn't brand new, right? There was something called the disdifferentiation theory way back in the 80s.
You're right. Richard Cutler proposed that back in 82. So yeah, the basic concept has been around for a while.
Okay, so the seed of the idea was there, but the ITOA is like a more, fleshed out modern version.
Exactly. The modern ITOA, especially the way David Sinclair is presenting it, it digs into more specific mechanisms and has more evidence to back it up.
Makes sense.
Oh, and one more thing. It's important to keep in mind what Aubrey de Grey pointed out. The ITOA is focused on the why of aging, like the root cause. But a lot of these exciting results with epigenetic reprogramming, that's more about how we could possibly intervene, how to fix it, not necessarily about proving the root cause. So they're related, but they're distinct questions.
Oh, I see. That's important. So it's not just about proving this theory. It's about, hey, even if it's not the cause, can we still do something about it?
Yeah. And then there's also, the scientific community generally agrees aging is complex. It's not just one thing. It's probably a whole bunch of factors all tangled up together. It's not likely that any single factor is responsible for more than maybe 50% of aging.
Okay, so aging is this huge puzzle and we're still figuring out all the pieces. But this idea of epigenetic information getting degraded, that's one of the key things David Sinclair is focusing on. So what kind of evidence is he presenting? How does he explain this noise accumulating and messing things up?
Right, so a big part of Sinclair's work is this idea. The main driver of aging in mammals is this lifelong accumulation of epigenetic noise. It's not just random noise, though. He proposes a specific way this noise happens, and it's actually linked to something essential, DNA repair.
Wait, DNA repair is causing epigenetic problems. That sounds kind of backwards. I mean, fixing damage should be good, right?
It seems that way, but his hypothesis is really intriguing. Basically, when your DNA gets damaged, let's say a double-strand break, which is a pretty bad one, the cell has to jump in and fix it. But during this repair process, especially when the DNA building blocks are actually being replaced, there's a chance of losing methylation, which is an important epigenetic mark right around the damaged spot.
I see. So it's like fixing one problem might create another.
Right. And it gets even more interesting. The proteins that normally shield certain parts of the genome, they might get pulled away to help with the repair. That leaves other areas exposed and vulnerable to, more damage.
So it's like having a mechanic fix your engine but accidentally scratch the paint while they're doing it.
Yeah, that's a perfect analogy. Now, Sinclair takes this even further and links it to Claude Shannon's information theory, which is fascinating. Shannon was all about how to send information reliably, even with interference or noise, messing things up. He came up with this idea of an observer, someone who has like a clean copy of the original signal, a backup, to fix any errors in the received signal.
So are you saying our cells might have a backup copy of our youthful epigenetic settings, like a restore to factory settings option?
That's exactly what Sinclair is suggesting. He thinks that when these errors build up in the epigenome, there might be some internal reader in the cell that can go back to this pristine backup and, reset the instructions. And that's what epigenetic reprogramming is all about.
Okay, this is getting really interesting. I've heard of those Yamanaka factors. Do they play a role in this whole cellular restore idea?
You got it. The Yamanaka factors, those are usually a combination of proteins like OCT4, SOX2, KLF4, that's OSK for short. Sometimes they add micardic to it, making it OSKM. What they do is reprogram adult cells, the specialized ones, back into something like stem cells, like induced pluripotent stem cells, which are more youthful and flexible. And the really cool thing is there's growing evidence that if you use these factors in a controlled way, just for partial reprogramming, you can actually reverse signs of aging.
So it's not like turning a skin cell completely back into an embryonic stem cell, which could be dangerous, but just like nudging it back to a younger version of itself.
Exactly. And there's some really amazing research on restoring eyesight in mice that illustrates this. That's highlighted in some of the stuff we've been looking at. They use gene therapy to deliver OSK to the retinal ganglion cells, those are the neurons in the eye, in both old mice and mice with glaucoma. And the results were amazing. They got their vision back.
Wow. That's incredible.
It is. And it wasn't just some surface level fix either. The treatment actually restored the transcriptome, which is all the RNA molecules in the cell, and the DNA methylome, the pattern of DNA methylation, back to a more youthful state. It even improved the function of those cells and their ability to regenerate.
So they reversed aging in the eye and treated a disease at the same time. That's a huge result.
It is. And here's another important finding. This effect actually depended on two enzymes, TET1 and TK2. They're like molecular erasers, getting rid of those misplaced epigenetic marks, which supports the idea that manipulating these DNA methylation patterns is key to this whole rejuvenation thing.
Okay, so there's some really solid evidence there.
There is. And another important thing, they did these studies where they delivered OSK throughout the bodies of mice for long periods, like 18 months in one study, and they didn't see a noticeable increase in tumors or other major bad health effects. That's always a big concern with these kinds of reprogramming approaches, right? You don't want to accidentally trigger cancer.
Oh, absolutely. That's always the worry when you're messing with how cells fundamentally work.
Yeah. But the fact that they didn't see that increase in cancer, that's really reassuring. And the exciting thing is it's not just eyesight. There's evidence for rejuvenation in a bunch of different tissues. Mm-hmm. Kidneys, liver, skin, heart, brain, pancreas, muscles, fat cells, even mesenchymal stem cells. And we're seeing not just changes in gene activity, but also in histone modifications, which is another level of epigenetic control.
Wow. So it's not just a one-off thing in the eye.
Nope. It seems to be more widespread. There's even this one study that used AAV-OSK gene therapy, and it actually extended the lifespan of older mice. And then there's this whole other area of research looking at using cocktails of small molecules. They target the epigenetic regulators to get similar rejuvenation effects. So lots of interesting stuff happening on that front, too.
So this is all pointing to a pretty optimistic future, right? The idea that we could actually reset our cells' epigenetic clocks. It's really compelling. But you mentioned earlier that not everyone's convinced. Let's switch gears now and talk about Aubrey de Grey. He's a big name in this field, but he seems to be a bit more cautious about all of this.
Yeah, de Grey makes a really important distinction. He separates theories about why we age from strategies about how to intervene. So he's acknowledging those cool results with OSKM, but he sees it more as a potential tool for repair, not necessarily proof that losing epigenetic information is the primary cause of aging.
So even if we can reverse some age-related changes, it doesn't automatically mean that epigenetic decay is the root of the whole aging process.
Right. And he uses this analogy, I think it's pretty helpful. He describes OSKM like a baseball bat. It just hits the entire genome, no discrimination. His point is, these interventions can't tell the difference between epigenetic marks that are just noise, the errors we want to fix, and the ones that are essential signal, the instructions that tell the cell what it's supposed to be. OSKM just removes a portion of both.
I see. So you might be messing with important cell functions while trying to clean up the mess.
Exactly. De Grey's argument is, if partial reprogramming actually works, it shows how tough and redundant a cell's epigenetic state is. Even after you mess with it quite a bit, it mostly keeps its identity.
That's interesting. So the cell has this strong underlying identity, like a core, that can handle some level of epigenetic tweaking.
Yep, that's the idea. But his biggest worry, the thing he really emphasizes, is the potential for cancer. He points out that as we age, we all accumulate cells with mutations, some of them the kind that could lead to cancer. Usually the body keeps these precancerous cells in check, but De Grey's concern is that using a baseball bat like OSKM on these cells might accidentally push them over the edge, make them fully cancerous.
And he's saying that short-term studies might not be enough to guarantee the safety of these reprogramming methods.
Exactly. It takes time for cancer to develop. You might not see the full consequences of these interventions in a short study. He's really pushing for long-term research to figure out the actual cancer risk. He's not saying partial reprogramming is a bad idea, but he's saying, hey, we have to nail down this cancer risk, and that's going to take time.
That makes a lot of sense. Safety first, right? Okay, so we've talked about Sinclair's work and De Grey's concerns. Now let's move on to Charles Brenner. His criticisms seem to be more about the actual experiments used to support ITOA, especially this one study that involved causing DNA damage on purpose.
Yeah, you're right. Brenner and James Timmons, too, they've raised some serious questions about how a specific study was interpreted. This study used an enzyme called IPPOI to create double-strand breaks in the DNA of mice. The idea was to show that this DNA damage, and then the repair process, would lead to premature aging, like what the ITOA predicts.
Okay. And what exactly is the issue with this IPPOI experiment, according to Brenner?
The main problem they're pointing out is that previous research, some of it even from Sinclair himself, had already shown that this IPPOI, it's mutagenic, meaning it can cause mutations, it's cytotoxic, which means it kills cells, and it's progeric, meaning it induces premature aging. And the reason it does that is because it causes cells to die off.
Oh, so they're saying that maybe the premature aging they saw in this study wasn't actually because of faithful DNA repair messing up the epigenome, but just because the enzyme they were using is known to be harmful and kill cells.
Yeah, exactly. They're saying the researchers might have jumped to the wrong conclusion because they didn't properly consider this other explanation, the fact that IPPOI is already known to be bad for cells.
That's pretty important. So why weren't they more careful about that?
Well, they're pointing out that in these older studies where IPPOI was targeted to specific cell types, like in T cells, the researchers saw a lot of cell death, like a really significant loss. And in epidermal stem cells, IPPOI made these cells almost disappear completely, which led to hair graying and thinning. And even the original researchers said that was because the cells were dying off. This was a cell elimination mechanism of aging.
Wow, so it's a pretty well-established thing that this IPPOI can cause a lot of damage and cell death.
Yeah.
And Brenner and Timmons are basically saying, hey, why wasn't this considered more seriously as an explanation in the newer study?
Exactly. They also point out that in this newer study, they used a drug to induce IPPOI expression all over the mice's bodies for 3 weeks. And they're arguing that doing it that way throughout the whole body would probably cause even more cell death than in those older studies where they targeted it more specifically.
I see. But did they even look for that cell death in the study?
That's the problem. The study didn't include any analysis of the mice's tissues in the first month after they stopped giving the IPPOI drug, and that would have been the most important time to see this potential wave of cell death based on what we know from previous research.
So they missed the window where the most obvious signs of damage would have been.
Yep. They only did their molecular analysis a month after stopping the drug, by which time most of the dead cells would have been cleared away by the body. So Brenner and Timmons are saying this creates a survivorship bias. The cells they analyzed were the ones that survived the IPPOI, not necessarily representative of what was happening when the damage and repair were actually going on. They actually asked for data on DNA damage response and cell death from those first 30 days, but they didn't get it. So they think that crucial part of the study might not have been looked at carefully enough.
So there might be some holes in that evidence then.
Yeah, and there's more. They're also saying the study didn't have proper controls. They didn't really compare their results to what would happen if cell death and inflammation were the main drivers of aging in this experiment, instead of those subtle epigenetic changes they're saying were caused by faithful DNA repair.
So the way the experiment was set up makes it hard to be sure what really caused the premature aging.
Exactly. And they also point out that there's no data showing that OSK treatment can actually reverse the specific aging features that were seen in this IPLPY study. They're saying, okay, you showed that OSK can rejuvenate things in general, but did it fix the problems caused by IPPOI in this model? And the answer is, we don't know, because that data isn't there.
Interesting. So all of that together, it seems like Brenner's critique is saying that maybe the link between faithful DNA repair and epigenetic aging, at least in that particular study, isn't as strong as it seems because of the problems with how the experiment was done and the lack of data from that critical early period.
Yeah, that's a great summary. And they're skeptical of the conclusions of that study given all these issues with the methods and the fact that the researchers didn't even reference their own earlier work that was relevant to this.
I guess it shows that even in cutting-edge science, you can still have these big disagreements about what the results really mean.
Absolutely. And, going back to what we talked about at the beginning, the ITOA is a very appealing idea, right? The possibility that aging has a reversible cause, that's exciting. But as our sources show, right now, it's still a theory that needs more evidence to back it up. The potential of epigenetic reprogramming is huge, no doubt. But we have to take those safety concerns seriously, especially the cancer risk. That's not something to mess around with.
Makes sense. And it's a good reminder that throughout history, scientists have always debated and even, had rivalries. But those different viewpoints, those arguments, they've actually helped us understand complex stuff better in the long run.
That's so true. Science is all about testing ideas, challenging each other, and being critical. That's how we get closer to the truth, really understanding how things work.
Okay, so let's recap. The ITOA, the information theory of aging, it's saying that what really drives aging is this gradual loss of epigenetic information in our cells. David Sinclair's research, especially the work with restoring eyesight in mice, suggests that we might be able to reverse this loss. He's talking about partial cellular reprogramming, using things like OSK to kind of reset the clock, so to speak. But then you have people like Aubrey de Grey who are raising really important questions about unintended consequences, like maybe triggering cancer by using these broad interventions. And then there's Charles Brenner, who's digging into the actual experimental evidence, finding some weaknesses in those studies that were supposed to be linking DNA repair to epigenetic aging.
Yep, you nailed it. That's the lay of the land right now. But keep in mind, it's a fast-moving field. There's always new research coming out.
It's exciting to see where it all goes. But for everyone listening, I think this deep dive gives us a really good overview of a really important area of research. I mean, this stuff has huge potential implications. And it really shows how complex aging is and how scientists are still trying to figure out all the pieces of that puzzle.
Right. And I think it's important to remember that even the most promising ideas in science still have to go through this process of being tested and debated.
Totally. Well, I hope everyone enjoyed this deep dive into the information theory of aging. This is just a starting point, though. If you're interested in learning more, we encourage you to go and check out the research we talked about and keep an eye out because this is a rapidly evolving area. There's always new stuff coming out.
Absolutely. And to leave you with something to ponder, if this idea of a cellular backup of youth is correct, what are the most crucial mechanisms that keep that information intact? And how can we safely and effectively use that knowledge, not just to live longer, but to live better, to be healthier and more vital as we age? That's the real question, isn't it?
That's the million-dollar question. Okay, well, thanks for joining us, everyone. Until next time.
See you next time.